There we go. All right. So yeah, today I'm going to be talking with you guys about heroin self-administration and abstinence in the ventral pallidum and nucleus accumbens, and talking about distinguishing subtypes in these regions. So first, a little background about this system. The nucleus accumbens and the ventral pallidum are central nodes in the mesolimbic dopamine system, which is an important part of the reward and motivation system. And this system mediates rewarding properties of addictive substances, and it undergoes lasting changes in opioid use disorder. The diversity of neuronal subtypes in these regions are incompletely characterized, particularly in the ventral pallidum. And the mechanisms by which drugs alter the activity of these neurons is still poorly understood. So we are using single-cell genomics to characterize these cell types and the changes which occur through heroin self-administration and abstinence. So this is our experimental design. Rats were trained to self-administer heroin or saline 3 hours daily for 10 days. And we conducted both RNA and ATAC-seq on samples. So we got samples from both the nucleus accumbens and ventral pallidum of each rat. We had 40 rats altogether in the scheme that you see here. And we took time points at 24 hours and 14 days of abstinence in both males and females. So first, I'm going to talk with you guys about the cellular diversity in the ventral pallidum. The ventral pallidum is the meeting place of a lot of different regions of the brain, and it's pretty diverse. On the right here, you're looking at all of the cells which passed our initial filtration of the single-cell RNA-seq data. And I'm going to be zooming in a little on the neurons that are sort of lassoed here over in this area, which we reclustered once we subset them out. And so we find a few different populations that I'm going to go into a little more on the next slide, but a large population of GABAergic neurons, a couple populations of medium spiny neurons, which there's a almost certain chance that a good portion of that is contamination from the adjacent nucleus accumbens, and then populations of both VGlut1 and VGlut2 glutamatergic neurons, and then a healthy population of interneurons as well. So looking at the expression of these different populations, we can see that there's a pretty robust population of both of those medium spiny neurons, like I mentioned. But in recent literature, it's been indicated that inside of the ventral pallidum, there is a population of D3 expressing medium spiny neurons, which I'm going to be coming back to later in this talk, because I believe that some of the D1 medium spiny neurons that we're looking at are playing an important role in the response to heroin use in the ventral pallidum. So first, I want to share some differential gene expression analysis on the 24 hours abstinence time point. So this is just a volcano plot showing that our differential gene expression is coming out looking like it should. And over here are a few genes that in at least one of the cell types seen here had a p-value of 0.05 and a false discovery rate of less than 0.01. And so this first gene is prostaglandin synthase, which synthesizes a neuromodulator, and it's generally upregulated across all of our cell types. So there's an increase of that neuromodulator in all of our neurons in the ventral pallidum. SOX5 is traditionally a developmental transcription factor, but we're seeing that there's a distinct upregulation. Sorry, the red is, if the red is taller, that is downregulation in response to heroin, and if the blue is taller, that is upregulation in response to heroin. So this is an upregulation here, and a downregulation here. An upregulation of SOX5 in certain populations, like our glutamatergic and non-medium spiny neuron GABAergic cells. And then what you're looking at the bottom here is a long non-coding RNA that has been shown in the literature to, when it is overexpressed, it leads to an increase in neuropathic pain, which matches the responses that we understand occur with heroin use, prolonged heroin use. And so we see that it is being generally upregulated in response to heroin use. Now I'm going to share 14 days, our 14 days time point with you. And as I go into it, I want to let you know that when I ran a Pearson correlation on the differential gene expression of 14 days and 24 hours of abstinence, it was a 0 correlation. So we're seeing completely different patterns when we go and we look at the gene set that's differentially expressed in our 14-day time point than in our 24-hour time point. So the genes that I'm sharing with you here, I realize that they're stacked sideways now instead of up and down, but this first gene is, it's a nicotinic acetylcholine receptor modulator. And so when it is expressed, it contributes to inhibiting calcium influx. We see that that is being upregulated in certain cell types, sort of generally upregulated, but more so in certain cell types. Then this zinc finger here is linked with body dysmorphic disorder currently in human literature. And it seems to have a upregulation in certain cell types stronger than in other cell types. Then somatostatin is being downregulated in most cell types across the 14-day time point. And lastly, FOXP2, a developmental transcription factor, which has been shown to be an important cell type delineator in the globus pallidus, is being downregulated in certain the oxytocin and D1 medium spiny neurons that we're finding in our data set. So I could go for days about all these individual genes that I can pick out, but it doesn't really help us get at the bigger question of what are the patterns of transcriptional changes which are occurring. So from there, we conducted k-means clustering across all of the cells that we have across the treated versus the saline versus heroin treated cells. And so I'm going to be zooming in on a couple patterns that I observed specifically in our D1 medium spiny neurons when I ran this k-means clustering. And this is sort of fresh out of the oven, so if it's a little rusty or a little rough around the edges, sorry about that. But these are just 3 of the patterns that came up. And when we go and look at our D1 medium spiny neurons, which are these lime green neurons here, and also we have our D2 medium spiny neurons, which are a smaller population here, we can see that there are some patterns of gene transcription which are changing more specifically in D1 medium spiny neurons than in D1 and D2 medium spiny neurons. So I'm excited to look at those because I believe that one or multiple of these patterns, 8 and 18, stand out to me more than 16 because it's present in our D2 medium spiny neurons that don't express DRD3. I think that one of these is representative of the D1 medium spiny neurons expressing DRD3 that are present in the ventral pallidum in other literature. And I'm excited to see how they're changing. And one last thing about the ventral pallidum here. This is just a sample of neuromodulators that are being distinctly expressed across 48 different clusters that we found. This is the highest resolution of clustering that we've generated thus far for this data set. And we are going to be taking these neuromodulators among other genes and running cosmics to identify which of these cell types are actually coming from the ventral pallidum versus which are coming from adjacent regions in order to get a better representation of what is actually changing in the ventral pallidum for our data. And now I'm going to share just briefly with you a little bit about the Nucleus Accumbens data that we're working with. So I'm doing something similar here where I'm just pulling out all of our neurons, and that's what you're looking at on the right here. We have rich diversity of both D2 and D1 medium spiny neurons. Down here, the distinguishing gene between our atypical 1 and atypical 2 is GRM8, but that also leads to a difference in glutamate receptor expression. So I think that this is going to be an interesting pattern to look at moving forward. But what I want to show you here is that with the help of Brian Herb, we have taken this data set and integrated it with the Kellis Labs data from the human Nucleus accumbens. And what we can see is that there's a really nice overlap between most of these cell types, which is very encouraging. That what we're looking at in changes in the rat should have a good ability to translate into changes that we would see in humans. There are some populations which seem to be a little more independent, like our cholinergic interneurons. Don't seem as well represented in the human data set. We'd have to look more into why that is, but all in all, we're really excited that this is matching up so well. And with that, I'd just like to say thank you to my whole lab, the MENT lab, and also the Dietz lab and the Loba lab, who I've been working closely with, and Scorch, University of Maryland IGS, and Mind, and the 3 grants that have been helping me from the NIH. And these are references, so thank you.
Great. We have time for a couple of questions, and we have one in the Q&A. So the excellent study, the graphs are very informative. Could you mention the software used to analyze your single-cell RNA-seq data?
Yeah, sure. I used Seurat primarily, and with the Canon smoothing, that was a package that I got from a member of my lab. I can definitely share that with Scorch. It has a README file about where it came from, but I would be happy to share. Most of the tools came from Seurat and Edjar. So. Sorry, really.
Thanks, Ben. Any other questions for Ben? Oh, we have one. Go ahead. You should be able to talk.
Hi, very nice data. I just was very intrigued by you finding the link RNA that is showing some changes. But as you know, finding link RNA, the single cell RNA data is very hard because of the way that are essentially expressed at very low levels. So I'm just wondering, because in our hands, at least, you know, the RNAs should be having a certain copy number before the results are reliable. So I'm just wondering if you have checked the copy number of this gene, the link RNA in bulk RNA-seq, to know how high the copy number is and how reliable so the single cell RNA-seq is. Thank you.
Yeah, with that link RNA is what you're specifically asking about? Yes,
exactly.
Yeah, we haven't taken a look in bulk RNA yet, but I will say that most of the genes that I was showing were on the higher end of counts per million reads in the data set. So this was probably coming up in the sequencing data at least 6 or 7 counts per million, which is on the higher end for our data, because nuclear data is a little sparser. So. But it would be really good to follow it up in bulk data, because the only literature that I found was an effort where they were specifically upregulating the gene with a viral infection. So.
Thank you. Something that has helped us a lot is doing a converting single cell RNA-seq to pseudobulk. That sometimes helps. Thank you.
Yes, this is pseudobulked. Yes, sorry, I should have mentioned that. The EdgeR analysis that I've done here is pseudobulked. My mistake. The KNN smoothing is at a single cell level, but this differential gene expression here is pseudobulked. Yep.